So I'm going to provide an overview of what we are doing, what we are planning for the future. Right. So then a few words about my interests and motivation. I'm fascinated about hidden biodiversity. I like to play this slide as a small play with the audience. I don't know how it's working now in online form. Basically, what you are seeing here is the number of eukaryotic species described and expected to exist, undescribed species, in millions. The length of the bars are showing the number of the described and undescribed species. Dark brown is everything what is known. Light brown is what is suspected that is out there, but we did not name it yet, did not find it yet. And next to the bars, on top of the bars, you could see the names, what groups these bars are referring to. And I would like to ask you if you could just guess what these different bars are. And I guess students are in advantage and can guess which eukaryotic organism group might be represented on Earth with many millions of species, and most of these species are not described. Well, I guess guessing online is difficult. I show it right away. So the first group is insects and myriapods. Not really surprising. There are estimated up to 10 million or even more species to be present, and around 1 million are described. There are many fungal species which are expected to exist and very few described. There are many chelicerates, mostly because of the mites, where one expects that there are millions of 1 million species undescribed. Basically, what we see on this graphic is that most of the biodiversity of Earth is not really known to date, and this is quite surprising, given the fact that there is a road in every kilometer if you are traversing the surface of the Earth. So how can it be that we find, we describe so few species, given that humans are kind of inhabiting the entire Earth and impacting all the Earth? So basically, this is because most of these undescribed species are small, are not ubiquitous, are difficult to see, and they are the ones which are fascinating me, and most of my research is focusing on methods and approaches to find them, and also to understand their ecology, their adaptations to the environment. Right, so I'm coming from the LOEWE Centre for Translational Biodiversity Genomics. I say a few words about this. This is a research project which started in 2018 with the support of the Hessian Ministry for Science and Arts. It is currently a cooperation of 6 institutes, 2 universities, the Goethe and the Justus Liebig University, the Senckenberg being the main participant in the project, 2 Fraunhofer institutes, and the Max Planck Institute for Terrestrial Microbiology from Marburg. The project is running until 2024 and is going to be incorporated into Senckenberg and several other partners after this. So LOEWE TBG works on biodiversity genomic research with high societal relevance. We are looking into loss of biodiversity, population growth, needs for food, better health, more resources, environmental issues. And we are looking into genomes to try to find solutions for all these problems by documenting genomic diversity through the project area comparative genomics, using genomic biodiversity, looking for and discovering natural products by genomics, and synthesizing these products and testing them, and protecting biodiversity through functional environmental genomics, genomic biomonitoring, which basically means that we are looking into opportunities provided by new genomic developments to perform sustainable conservation and management of natural resources. All right, so my talk is structured in two parts. The first part, the longer one, is about the functional environmental genomics of soil invertebrates. And the second one is about generating ecological time series with environmental DNA. So I'm breaking up the talk into these two parts because these are basically my main research interests and the directions I'm developing in the next years. So, yeah, so soil biodiversity, what you see here on the spoon is a few grams of soils. And you can find a tremendous diversity in this 1 gram of soil. So again, I would like to play a guessing game here, but I'm not sure if this is going to work again. In this 1 gram soil, you might find about 200 meters of fungal hyphae. There also might be like 1 billion bacterial cells. And I would like to ask you to guess how many species of soil invertebrates there might be present in 1 gram soil. You could also, I mean, chat is running. Insects, thanks. Oh, okay, great, thanks. Okay, I discovered this wealth of answers in the chat now. Great, cool. Guesses are going up from 100 from Astrid to 5,000 from Julia Schwarzer. And I just revealed this now. It is several thousands of taxa. It is, of course, very much dependent on what kind of soil we are looking at. But if you are looking at soils which are very rich, organic matter and so on, they can support several thousands of soil invertebrate taxa. And I guess this is a surprising number. It was a surprising number for me, given how small amount 1 gram is. Now, these are the main groups of soil invertebrates. So these are the really small ones. That's why you have such a huge number of species in basically most areas of Earth. These are very small invertebrates, collembola, nematodes, all kinds of mites, small worms like this anchytrid worm here. And all of these species have very interesting ecology and evolution. They're adapted to a strange environment, which is full of microbes, has challenging conditions, drought conditions, humidity conditions, pH, and so on. They possess strange adaptations. For example, they can synthesize weird natural products that you would not expect from animals. For example, springtails. Many species of springtails are known to be able to synthesize penicillin. Antibiotic synthesis was not found by any other animal group so far. They also have weird sexuality. They are hermaphroditic species. There are many parthenogenetic species. Very often, species pairs can be sexually reproducing or entirely parthenogenetic. And all of these species are contributing very much to soil functioning and health. And that's something I find very important. That's the reason why I decided to start working more on soil invertebrates. So my aims with the functional environmental genomics of soil invertebrates is to create genomic resources which allow to understand the biodiversity of these fascinating small organisms better, and to evaluate new methods like metagenomics, metatranscriptomics, soil eDNA work, evaluate these methods to describe community composition better, and to understand how environment and species interactions are shaping the communities and ecosystem functions of these organisms. And obviously, this is in part very theoretical, very basic science, very curiosity-driven research. On the other side, it has tremendous application potential because soil management is very much needed. Major international organizations are speaking up for better soil management, and basically the methods which would allow to monitor soil biodiversity are just not up to the task of dealing with this tremendous biodiversity that we are finding in the soils. We just simply don't have enough people who can identify these species, and it's very time consuming to work with these communities. All right. So now the next couple of slides are about a project that I started with within TBG. This is the MetaInvert project, the metagenomic monitoring of soil invertebrate communities. That's a work that I started with Ricarda Lemitz, who is curator of the Oribatid mites in Görlitz. Many of these colleagues who are participating in this project were or are working in Görlitz in the soil zoology division. Currently, they are still Karin Hochberg working on nematodes, and Clement Schneider, who is a curator of Apterygota. And other colleagues are helping us with enchytrates, species identification, sorting, myriapods, and kilopods. This is a project which would not be possible without this intensive collaboration with a very broad taxonomic expertise, a very strong taxonomic community who can actually help us identify the species that are out there, help us tell what to sequence, and how to set up experiments for metagenomics. Right, so the MetaInvert was conceived as a metagenomic monitoring approach. Metagenomic, just as metabarcoding, needs databases. But while metabarcoding is needing barcode databases, metagenomics needs genomes. So we set up this project to actually genome sequence many soil invertebrate species and use these genomes for supporting metagenomic identification. This is reflected by the sampling of the taxonomic groups which we sequence. The colleagues from the soil zoology are providing us with broad coverage of taxa from the most important groups of soil invertebrates. So we are sampling very broadly in Colembola, in mites, including Oribatid mites, in nematodes, in diplopods, and many other species. Our aim here is to generate a taxonomically broad coverage genome database. And it was already clear when we started the project back in 2018 that the recent advances in genome sequencing allow us to actually not go for barcoding these species, but rather go for shallow genome sequencing. Now this changed to date, and we could go even better. We could produce draft genomes, higher coverage genomes for many of these species. And I'm just giving a quick overview of what we got to date. We were performing over 500 genome sequencing reactions in the last couple of years, and managed to produce genome sequences for around 250 species of soil invertebrates. When the project was first spearheaded by Clement, who by that time was doing a postdoc with me, starting on this project, and then he left to become the curator of Apterygota in Görlitz, and then Gemma Collins was picking up this project and leading the analysis of these genomes. So most of these soil invertebrate genomes were done from single individuals, except the nematodes, which are mostly very small, and there we are pulling several specimens from cultures. They were sequenced to a fixed depth of about 10 gigabase each species, and they were assembled. Microbiome sequences were identified and removed from the genome sequences, and the rest of the sequences were used to construct phylogenies, analyze a couple of genome traits, and to correlate these genome traits with ecological traits. We are looking into habitat generality of these species, how this is the reproduction mode of the species, body size, and how all of these are interacting with genome properties. what you're seeing on this on the circle here, this is basically a phylogenetic tree of these 250 genomes based on single copy ortholog genes, and completed with a couple of genomes from GeneBank to kind of place the genomes properly into the phylogeny. Everything what is colored is genomes that we are providing for the community now. Largest part of these genomes are coming from springtails. The second largest groups are oributid mites. And we are also focusing on a couple of other groups here. Right. Oops, that doesn't look like very good resolution. Sorry for that. What we are finding is that there is a tremendous variation in the genomic traits. What I'm showing here is a graph from JEMA, which is on one hand shows the same phylogeny now in an extended format, and shows the genome size in those groups for every species for which we have a genome. These genome sizes are estimated with a new tool that my colleague Markus Fenninger has published a couple of months ago. It is based on coverage distributions, and he could show that this genome size estimation tool is providing basically a one-to-one genome size estimation compared to flow cytometry, and it is considerably better than k-mer-based genome size estimation approaches. So what we see here is that genome sizes vary a lot among the groups, but also vary a lot within the groups. The among group variation is about 30-fold. The smallest genomes are around 70 megabase in nematodes, and the largest genomes that we are finding are almost 3 gigabase in one diplopod species. What is surprising is the range of variation in genome size within groups for which we long consider that genome sizes are relatively small, for example, the oributids, but also the collembola. We see similar variation in traits in GC content and effective population size as well. And then we are working on to identify the relationships between the genomic and ecological traits and see whether these relationships are conforming predicted theoretical relationships from genomic and evolutionary theory. So on this graph here, which is this graph here is the basis of a structural equation model where JEMA was connecting ecological traits with genomic traits. And each of these arrows are conforming to a theoretical expectation between those traits. So I will just quickly walk you through on this graphic. One important expectation is that I start here with arrow 1 here, which basically connects repeat content to genome size. We expect that repeat content is one of the most important factors which is driving genome size. So we expect a strong positive correlation here. Body size, at least in some assumptions, should also influence genome size positively, because very often body size is related to larger cell size, and cell size is very often related to a largest genome size. This holds true for some organisms, but not for everything. This is something we know from previous works. Body size should be connected to effective population size, which is something that we can estimate from genomes. Large body organisms tend to have smaller effective population sizes and vice versa. Effective population size, on the other hand, should negatively influence genome size and repeat content, because we would expect that selection in larger populations is more efficient and can remove unused parts of the genomes more efficiently, contributing to reduced repeat content and smaller genome sizes. We also expect that habitat generality is positively linked to effective population size. Basically, species which are present in more habitats should have larger populations. Reproduction mode and whether an organism is cultured or not obviously should influence effective population size. And then what we are finding in the genome data, once we are correlating the genomic and ecological traits, is that there are very few consistent patterns across the taxonomic groups. The most obvious consistent pattern is the relationship between repeat content and genome size. So it would have been strange if you don't find this one. It looks like in every organism group, if the genome contains more repeats, then genome sizes are just larger. But I think it's important to note that there is a considerable variation in the slope of these curves. So there is a lot of difference between the taxonomic groups. Right. And then if you're looking into other relationships of traits, we find that the taxon groups tend to behave very differently. One relationship that we expected to see is that the GC content should be negatively related to repeat content, because repeats generally contain more A and T. This is something that we are seeing largely in the data, but there are also several exceptions to this pattern. In some organism groups, it looks like the GC content is higher if the genome has a higher repeat content. When we're looking at the body size, genome size correlation, we find a very strong, very positive correlation. So far, we are talking about very small bodied organisms. In this case, larger the organism gets, larger the genome size is. However, this is plateauing off at larger body sizes. And then the trend is even reversing in the largest body size groups, and then those are diplopods and kilopods. There are several species which have large bodies but quite small genomes, and several species have rather medium-sized genomes but large, medium-sized bodies but larger genomes. All right, and this is kind of repeating with genome size and effective population size and habitat generality. What I was explaining all of this is that it's obviously we can have theoretical expectations based on the relationship of genome properties and ecological traits, but these relationships seem to be taxon specific. It looks like these relationships are not consistent across the taxonomic groups, and this is probably because of these groups having very distinct adaptations. All right, and another interesting result from here is that we are identifying quite a number of missing metazone orthologs in these small soil invertebrates. What you see here is a plot of ortholog presence in Collembola, myriapods, oribatid mites, and nematodes. Ingo Ebersberger and Hanna Mühlbayer from the Goethe University in Frankfurt, they were performing an FDOG assembly-based ortholog identification in these draft genomes. This is a new tool which can deal with draft genomes and possibly missing genome fractions. They were basically checking whether 1,500 metazone orthologs can be found in the soil invertebrate groups. And what we are identifying here is quite large numbers of genes that are consistently missing from springtails, sometimes from myriapods, from oribatid mites, and not that much from nematodes. We don't know too much about the function of these genes. It is very difficult to functionally annotate these genes because simply soil invertebrate gene functions are fairly poorly known, and what we can use for annotation, they are very distantly related taxa. All right, so quickly sum it up what we have so far here with the Illumina genomes. We see large variation in genome traits within the taxon groups, but also across the taxon groups. We find only a few general patterns in genomic and ecological traits, but there are quite a lot of group-specific patterns. We find consistently missing orthologs, but we don't really know what those orthologs are doing. And altogether, in my opinion, what this is calling for is a need for broad and deep taxonomic sampling when somebody wants to perform comparative genomics. Basically, these patterns could not be identified and could not be picked up if if we are comparing only species pairs or only a few species from different taxonomic groups. We need this deep taxonomic sampling within the groups to be able to identify the outliers and look into the outliers. And those outliers are obviously present. All right. We are not only looking into Illumina genomes, but we are also interested in reference collected genomes for eukaryotes. Most of you probably are aware of the big international moves for generating reference genomes for all eukaryotes of Earth. Here I just mentioned the Earth Biogenome Project and two nodes, the ERGA and the African Biogenome Project. Recently, we published an opinion in 3 about why these reference genomes are needed in conservation and how these reference genomes can help with all kinds of conservation-related questions, from identifying inbreeding, outbreeding introgressions, but also in community biomonitoring. We also started a small genome initiative, a soil invertebrate genome initiative, with several colleagues from several European institutes. Karen Moiseman from ZFMK is also involved in last year. And our aim here is to produce more reference collected genome resources for these small soil invertebrates. What we did so far is we are trying to work with single specimen PEG biosequencing from springtails and oribatid mites and nematodes. The challenge with reference genomes and soil invertebrates is that these animals tend to be extremely small. So here are 2 species for which we managed to do high quality genomes, Desoria tigrina and Sminthurides aquaticus. You can see a millimeter scale here. Basically, you see that the Desoria is about 2 millimeter long, and the Sminthurides female, which is the larger sex, is almost 1 millimeter long, and the male is tiny. You can see the same species, the Sminthurides species in nature. The large one is the female coated by the really, really small male. So they are really small, and one of the biggest challenges is that they are not yielding large amounts of DNA. And so far, high quality genomes need a lot of DNA. There are issues with collection. There are issues with DNA degradation, because many of these soil invertebrates need to be field collected, then preserved on the field, very often in ethanol, and sometimes stored before they can be identified and sequenced. And there's also interspecific diversity, so we cannot just simply collect a large population and pool the specimens, because there are many pathogenetic species, and even if the species are sexually reproducing, there is a lot of interspecific diversity, even within single populations. So what we managed so far with these 2 species is, I'm just calling out the sheer numbers, because we were very happy to have those. We managed genomes with 4 to 8 megabase Contig N50 with the PacBio Ultra Low kit, with the help of Bruno Hütter from the MPGC from Köln. Very few numbers of contigs. So even without scaffolding, we go below 200 contigs and fairly complete genomes. So these are only 2 species, but we are putting a lot of effort to optimize these methods and get high quality genomes for more springtail species and more mite species. Leonie Schart, who is leading my lab in Frankfurt, is working on currently to optimize extractions and library preparation for nematodes, a bit of in collaborations with Philipp Schiffer from Köln. And Leonie managed to produce high quality genomes for quite a number of springtail species and mite species in the recent months. With these genomes, we want to look into the comparative genomics of adaptations to growth and land use gradients in species pairs. All right, so time is flying. I should also kind of speed up. So the second part, how are we using these genomes for metagenomics? What you see here is a quite typical soil invertebrate community extraction. All of those tiny little dots in the petri dish are single specimens. If you zoom on, you see that they are nice structures. They are mites and springtails, and diplopods, and other mites. But basically, it's very difficult to work with them because it needs a lot of knowledge to identify them, and it's also very time-consuming. So these kind of communities are kind of calling for DNA-based identifications. The obvious choice would be metabarcoding, which consists of sampling a community, extracting DNA from all the specimens together, amplifying a PCR marker gene, doing high-throughput sequencing, and then bioinformatics to get species identities. An alternative here is shotgun metagenomics, where we just extract DNA and do not amplify anything, but kind of high-throughput sequence the DNA as it is on a sequencer, and then do the bioinformatics. Why this is promising is that PCRs are suspected to contribute a lot of PCR bias and distort read biomass relationships. So basically, you get a number of reads, but those reads are not reflecting of how much biomass a certain species had in your original samples. And we are expecting that we can get rid of this bias by avoiding the PCR and doing shotgun metagenomics. Now, I just want to point out that soil invertebrates are excellent candidates for this because the way they are traditionally collected, what you see on the left, these are bellies, funnels with a soil sample inside. So soil is stored here, and then heat is applied from the top. The soil is slowly drying out, and the reaction of these microarthropods, at least, is that they quickly start to crawl down to more humid areas. And if we put a jar here with some preservation liquid, they just crawl in, and we can collect the jar and have a fairly clean, enriched sample of soil invertebrate individuals. And you can do this in large quantities. You can extract kind of hundreds of samples at once in large equipments like this McFadden extractor here. All right, so we were looking into how well actually reads and biomass relationships are reflected in metagenomics. The Görlitz colleagues compiled for us 4 types of mock communities. Each of these mock communities consisted of a large number of specimens from 28 soil invertebrate species. They were a bit different because we were trying to simulate different abundance ratios among the specimens. And each of these mocks were replicated 3 times. So you can imagine this is a tremendous work, given that, for example, in this community, this nematode here had to be represented by almost 2,000 specimens, while some other species were represented by a fraction of a specimen, like larger enchytrids. We shotgun sequenced this, and this was the master thesis of Alexandra Schmidt, when Sue is also around. So, hi, Alex. What we found is that biomass is actually related to the number of assigned reads. So on these graphs, these are scatter plots. On the x-axis, you see biomass and other parameters. On the y-axis, you see the number of assigned reads. With increasing biomass, we are getting more assigned reads. However, this relationship is not as strong as we expected. We also see a lot of taxonomic difference among the reads. So some of the groups, like the Colembula here, are getting much more reads from metagenomics than the enchytrids down here. We also see that the assembly properties and the genome properties influencing the read assignment. So, for example, species with higher genomes get more metagenomic reads assigned, which is not surprising, but it's a factor which should be considered if somebody is doing metagenomics. And this is reflected on how important these different variables are when we are looking at assigned reads. The most important factors by far is the taxonomic identity of the species. what was very nice from these studies is that we were doing quite deep sequencing of these metagenomic communities, which is fairly cost intensive. So we tried to actually evaluate how much can we go down with the sequencing efforts if we can use much less money to get similar information from the metagenomic sequencing. So we downsampled the replicates to about 100-fold to only a couple of several thousand reads, and we basically see exactly the same pattern. So this is a very good news. Basically, if we are interested to do taxonomic identification with metagenomics, we can go with fairly low sequencing effort. This effort is not much higher than for metabarcoding. All right, to sum this quickly up. We have now the genome databases available make it possible to do metagenomics, but very importantly, taxonomy seems to influence the sequencing success. We suspect that this is because of the DNA content. It looks like the DNA extraction efficiency is very different from across different groups. And this has also important consequences for metabarcoding, because in metabarcoding, very often it is suspected that it is the PCR bias which is influencing the biomass read relationship. However, the PCR bias is a taxonomic bias. So some species are better amplified by the metabarcoding PCR than other species. This taxonomic bias seems to be manifested also in the DNA content or the extraction efficiency. So we can expect more DNA and get more sequences from some species, but not from the others. And these two factors are collinear. So actually the PCR bias in metabarcoding can be to some extent or to a large extent a result of differences in DNA extraction efficiencies. And this can be only checked if somebody is performing a number of comparisons with metabarcoding and metagenomics. All right, so I see that I really badly underestimated what I need for this talk. So I very quickly screen through and try to get to the time series, because otherwise we have no time for that. But as a small introduction to the time series, I want to show very few results from a soil eDNA metabarcoding study, what we are also doing to generate environmental DNA time series. This is a study that we recently performed in the Rhine-Main Observatorium, which is an ELTE site very close to Frankfurt. Although metagenomics and like metatranscriptomics are kind of very interesting and exotic new tools that are becoming more prevalent in ecological research, metabarcoding has a tremendous advantage. It can process a large number of samples, and this means that we can look into questions which really need this huge sampling depth. One of these things is generating time series. So this is very often focusing on a very few spots in space, and then sampling these sites over and over in time, and reconstructing community composition to see if communities are changing over time. So what we did here is a bi-weekly sampling in the Rhine-Main Observatorium in a floodplain. We took soils, quite large compound samples, extracted DNA, did PCRs with these, with 4 markers, and we looked for eukaryotes, animals, fungi, and plants in this soil eDNA to answer a question whether we see the seasonal dynamics in the floodplain community. And quickly, just one result. This is a work that was actually analyzed by Juliana, who is coming from ZFMK, and I'm very happy to have her here and also in my group. This is a quick result, basically showing OTU richness in these floodplain samples. On the x-axis, you see the time series from spring or early summer of 2017 until summer 2018. And the y-axis, you see OTU richness. And what we are picking up is a quite nice signal in the seasonality species richness, OTO richness, actually, higher richness and higher variance being found in summer months, then quite low richness in winter months, and its richness going up again in the next summer. This is quite reminiscent of what people are finding in aquatic eDNA. All right, so now I switch to let's last few minutes to long ecological time series with environmental DNA. This is centered around human impact in the Anthropocene. What we know is that human impact was exponentially growing during the great acceleration during the last few decades. What we are seeing from all kinds of measures of human activity is that we increase our activities in every fields, like in water use, damming of rivers, using fertilizers. Human population is skyrocketing thanks to good health care. And in general, we are consuming many more resources. On one hand, this is a very nice thing. So we are, as humanity, living in a fairly nice times compared to earlier historical times. But this is putting also a tremendous pressure on biodiversity. We don't understand very well these pressures because we don't have very good temporal biodiversity data. And I quickly want to demonstrate this to you. This is a short movie which is compiled from species observation records from the Global Biodiversity Information Facility. All those red dots that you are seeing on this map are individual observations of species. So what you can see in the 2010s, there are large parts of Earth which are very, very strongly investigated for all kinds of species. Once I click, this movie starts rolling back in time, and we can see how biodiversity observations are disappearing as we go back to earlier times. So I click, and the time is rolling back here on the left side. Now, probably you can see already that some of the observations are disappearing and more areas of the Earth are becoming white. And as we are getting close to the beginning of the 20th century, we see that most of the Earth's surface becomes white, and we simply just don't have biodiversity data from there. So this lack of data represents a major problem for understanding how biodiversity is reacting to long-term human impact. Right. I don't want to say that there are no very good not very good long-term biodiversity records. I also kind of sum up here what we have for understanding biodiversity dynamics over the scales of hundreds to thousands and millions of years. I just kind of grouped typical ecological studies along two axes. One axis is a time axis. It kind of shows how long a study lasts, from a couple of days on the left side to millions of years on the right side. So this is the times that the study is covering, the time range. On the y-axis, this is the taxonomic coverage that ecological studies are covering, from one species to a few species to very many species. On the short time scales, we have standard studies. Those are typical ecological studies working on single species and single species reactions to all kinds of factors. Then meta-analyses are summarizing these studies. We also have biomonitoring and long-term experiments, which can go to timescales of decades and maybe up to 100 years. Then beyond this, we have quaternary paleoecological studies, which are addressing Pleistocene changes with subfossilized organisms. And on a very long time scales, we have paleontological studies which are using fossils to look into biodiversity dynamics on the scales of millions of years. What you can see is basically the taxonomic coverage is decreasing strongly as we are progressing in time. If you are looking into very old times, we have to look into species which are fossilizing well. And this is a tiny fraction of biodiversity. Most of the species just simply don't leave remains behind that we can actually identify. So all our understanding of very long-term biodiversity change is coming from a few fossilized taxa. Now, this is something that we can actually change with environmental DNA, because environmental DNA is quite well preserved in some archives, like in sediments. And we can use eDNA to get high taxonomic coverage and also to look back to timescales of hundreds, thousands, tens of thousands, sometimes even hundreds of thousands of years. And in this very last part of the talk, I try to kind of wrap it up soon. I show a few results and some perspectives. I quickly lead you through how this is working. In the case of sediment cores, what we are doing is we are taking sediment core samples, basically coring through the lake bottoms. That's how a sediment core sample looks like. What you can see here is a stratification. Different layers are deposited in different times. So lower layers are deposited in earlier time horizons, and then we are going toward the top of a core, we see more recently deposited layers. We can date these layers with interesting methods like cesium, lead, or carbon. We can use these horizons to reconstruct the environment from biotic and abiotic environmental proxies, elemental composition, and so on. And we can also use these horizons to extract DNA, sequence it, and understand the communities that we're living in these old times. Right, so this is a project that I was doing a couple of years ago in northeastern Germany. We were working on 10 lakes and wanted to reconstruct community change over a few hundreds of years. We did metabarcoding of all eukaryotes, and we're interested in how pollution and eutrophication is influencing these lake communities. We have a good taxonomic coverage because we were working with a number of taxonomic markers, quite large number of PCR reactions, quite large number of sediment horizons from these 10 lakes. We got a lot of taxa, and also the resolution that we get for these taxa is quite nice. So we have a lot of OTUs identified to different taxonomic levels. What we see here, and that's what I wanted to show you for sure, because these are the results from the eDNA time series part, is a couple of interesting patterns through time. On these plots, what you can see on the x-axis is the time scale going from earlier times on the left toward the present on the right, and changing variables on the y-axis. The first graphic shows the average success of PCR reactions. So PCR reaction success depends on basically two things. One is the availability of DNA as template for the PCR, and the other is inhibition, these PCR reactions. What we see is that there is quite a constant PCR success rate through most of this time, most of these centuries, kind of until the end of the 19th century, where we see a tremendous jump in PCR success. So this jump is very likely a consequence of increased DNA templates in the 20th century. This increase in DNA templates is very likely not a result of DNA degradation, and I can go into details if you have questions about that, but it's rather a result of increasing DNA production in these lakes. Basically, productivity and the general biomass in these lakes tremendously increased in the 20th century. And this is kind of in line what we know about eutrophication. This is reflected in the richness of OTUs in all of these lakes. We have quite a stable richness for hundreds of years, and then in the 19th century and very much in the 20th century, the species richness goes up a lot in these lakes. This can be because of the carrying capacity of these lakes, which increased due to the productivity change. Could be also because of the homogenization of taxa. We can see changes in community composition, and that's something what I'm covering here with two measures of compositional change. One is turnover rate, and the other measure is a turnover compared to a baseline. Turnover rate is basically compositional change between consecutive sediment horizons, consecutive time points. If turnover rate is high, then this means that compositional change from one time horizon to the other time horizon is very fast. If turnover is lower, turnover rate is lower, this means that the communities change between consecutive times less. What we see here is that turnover rates were quite high for the earlier time periods, and then they go down in the 20th century. And this might be in agreement with what you would expect under eutrophication, where a couple of taxa become well established and dominate these communities. These taxa are able to cope with high nutrient conditions and actually benefit from these nutrient conditions to outgrow others. The other measure is turnover compared to a baseline, where basically community composition is compared in every time horizon to an initial community composition. What we see here is that we again seem to have some kind of a baseline turnover where communities change a lot, but then this change increases and communities drift away very much from communities as communities were in the earlier centuries. In the same time, we see a lot of change in environmental drivers. Eutrophication is increasing along a similar pattern. Heavy metal pollution is increasing similarly, although starting somewhat earlier. DDT pollution is increasing and then decreasing later on. Right. So what you can take from this is that basically with this tremendously complex data set, we can see and distill a couple of general patterns of how ecological communities are changing over these long time scales. And what we see is that this community change starts well before the beginning of the 20th century. Turnover rates are changing in the earlier centuries, just as the drivers are. And then I wanted to say a few words. We want to follow this up now in the PhD project of Juliana, looking at the history and future of the biotic phytoplankton communities. Basically, we are taking several courses from the Baltics in the last year. Some of these courses are very long and go back the entire 8,500 years history of the Baltic Sea. And Juliana and Alex were extracting DNA from these cores, and we are going to go into community characterization quite soon. All right, so to quickly sum it up, we can record broad taxonomic diversity over large time scales with eDNA. We can see human impacts before the 20th century, and we can see the major change in the Anthropocene. And it also seems that we can also identify biodiversity baselines, especially in the rates of change before human impact. And we can do that on very long time scales. So sorry for being too late, too long, and too dense. Thanks a lot.